Thermoresponsive particle synthesis in microfluidic systems is an exceptionally dynamic field at the intersection of materials science, polymer chemistry, and microengineering. In these systems, smart polymers, materials that can change their physical properties in response to temperature fluctuations, are synthesized within microchannels that allow for unparalleled control over reaction conditions. At the molecular level, thermoresponsive polymers such as poly-N-isopropylalkylamide, PNIPAM, are designed to undergo reversible phase transitions at a critical temperature known as the lower critical solution temperature, LCST. Below the LCST, these polymers are hydrophilic, absorbing water and swelling, whereas above the LCST, they become hydrophobic, expelling water and collapsing into a more compact structure. This dramatic change in behavior can be harnessed for numerous applications, including controlled drug delivery, smart coatings, and sensors. In a microfluidic system, the synthesis process begins with the precise injection of polymer precursors, initiators, and crosslinkers into a network of microchannels. Due to the inherently laminar flow within these channels, mixing occurs primarily through diffusion rather than turbulence. This results in highly uniform reaction environments, allowing researchers to produce monodispersed droplets that serve as individual reaction vessels. Imagine a microfluidic chip as a miniature assembly line where each droplet is a tiny laboratory, meticulously engineered to provide identical conditions for polymerization. This level of control ensures that the resulting thermoresponsive particles have uniform sizes, shapes, and most importantly, consistent phase transition temperatures. One key advantage of microfluidic synthesis over traditional bulk methods is the ability to rapidly and precisely tune reaction parameters. For instance, the flow rates of the precursor solutions can be adjusted to control the residence time within the microchannels, which in turn affects the degree of polymerization and cross-linking density. Temperature control is equally critical. Integrated microheaters or external thermal baths can maintain or create gradients along the microchannels, enabling researchers to dictate when and where the polymer undergoes its hydrophilic to hydrophobic transition. You might ask, why is this level of precision so important? The answer is that slight variations in particle size or the LCST can lead to significant differences in functionality, especially in applications like drug delivery, where release profiles are intimately tied to particle behavior. During the synthesis process, the microfluidic environment offers an ideal setting for real-time monitoring. Advanced optical techniques such as inline fluorescence microscopy and dynamic light scattering can be employed to observe the formation and evolution of thermoresponsive particles as they are generated. For example, by incorporating a fluorescent tag into the polymer chains, researchers can monitor the change in fluorescence intensity as the polymer transitions at the LCST. This provides immediate feedback on whether the desired phase transition is occurring at the correct temperature. Real-time monitoring not only ensures that the synthesis process is on track, but also allows for rapid adjustments. If the observed LCST deviates from the expected value, parameters such as flow rate or reactant concentration can be tweaked in real time, much like adjusting the settings on a high-precision instrument to maintain optimal performance. Thermoresponsive particles synthesized in microfluidic systems have a broad range of applications. In controlled drug delivery, for instance, these particles can encapsulate therapeutic agents and release them in response to a specific temperature trigger. Consider a scenario where a patient receives a drug-loaded PNIP particle that remains stable at normal body temperature, but releases its payload when the local temperature increases due to inflammation or targeted hyperthermia. This targeted release minimizes systemic side effects and maximizes drug efficacy at the site of pathology. Beyond drug delivery, thermoresponsive particles are also used in fabricating smart coatings that adjust their permeability based on ambient temperature. Such coatings can regulate moisture or gas exchange, proving beneficial in agricultural films or packaging materials that require controlled microenvironments. Environmental sensing is another exciting application. Thermoresponsive particles can serve as sensitive detectors that alter their optical or physical properties in response to temperature variations. For instance, sensors based on these particles might change color when exposed to temperature extremes, providing a visual cue that can be used in monitoring industrial processes or even in consumer products. Imagine a sensor embedded in a building's facade that alerts maintenance personnel when temperatures reach a critical level, thereby preventing structural damage due to thermal expansion or contraction. In soft robotics, thermo-responsive hydrogels are used to create actuators that mimic biological muscles. These materials can contract or expand in response to temperature changes, allowing robots to perform delicate, adaptive movements. For example, a soft robotic gripper made from PNIPAM-based hydrogels could gently grasp and release objects by simply varying the temperature, offering a level of dexterity and sensitivity that is challenging to achieve with traditional rigid materials. The design of thermoresponsive particles also involves exploring multi-responsive systems. Researchers are developing composite particles that combine thermoresponsiveness with other stimuli, such as pH, light, or magnetic fields. These multi-receptive materials can offer a more sophisticated control mechanism. For example, a particle might first respond to a temperature change by altering its size, and then, upon exposure to a specific light wavelength, change its surface charge to facilitate targeted cell adhesion. Such dual or triple responsiveness is especially promising in biomedical applications, where complex environmental cues are often present. A common question in the field is what challenges must be overcome to scale up the production of thermoresponsive particles using microfluidics? While microfluidic systems provide exceptional control at the lab scale, transitioning to industrial scale production requires addressing issues of throughput and reproducibility. Researchers are tackling this by designing parallel microfluidic arrays that can operate simultaneously, ensuring that thousands or even millions of particles can be produced with the same uniformity as those synthesized in single-channel devices. Advances in microfabrication and automation are making it increasingly feasible to scale up these processes without compromising the precise control that is the hallmark of microfluidic synthesis. Furthermore, understanding the kinetics of phase transition in thermoresponsive polymers is a subject of intense research. The rate at which a polymer transitions from a hydrophilic to a hydrophobic state can be influenced by factors such as molecular weight, cross-linking density, and the presence of additives. Microfluidic systems enable detailed kinetic studies by allowing researchers to systematically vary these parameters in a controlled environment. For instance, by modulating the concentration of cross-linkers in a PNIPAM synthesis reaction, scientists can observe how the LCST shifts and how quickly the polymer collapses once the temperature threshold is crossed. Such insights are invaluable for tailoring the particles to specific applications, ensuring that they behave predictably under physiological or environmental conditions. The integration of computational modeling with experimental microfluidic synthesis is another exciting frontier. By using simulation tools, researchers can predict how changes in flow rate, temperature, and chemical composition will affect particle formation and behavior. These models help in designing experiments and optimizing synthesis conditions before physical trials are conducted, saving time and resources. Machine learning algorithms are now being applied to the vast data sets generated by microfluidic experiments, identifying subtle correlations between synthesis parameters and particle properties that might be missed by conventional analysis. This data-driven approach is accelerating the development of thermoresponsive particles with tailored functionalities. In addition, there is significant research focused on enhancing the biocompatibility and stability of thermoresponsive particles for in vivo applications. For biomedical use, it is critical that these particles do not evoke an immune response or degrade into harmful byproducts. Researchers are exploring various surface modification techniques, such as pegylation, the attachment of polyethylene glycol chains, to improve the circulation time and stability of particles in the body. For instance, pegylated PNIPAM particles have been shown to exhibit reduced protein absorption and prolonged stability in physiological environments, making them more suitable for drug delivery and diagnostic application. Moreover, the precise control provided by microfluidic systems allows for the incorporation of complex functionalities into thermoresponsive particles. One innovative approach is the fabrication of core shell structures, where the core might contain a drug or imaging agent. and the shell is designed to respond to temperature changes. These core shell particles can be engineered to release their cargo in a controlled manner when the temperature is raised, ensuring that the therapeutic or diagnostic agent is delivered only at the desired site and time. This is analogous to having a time-Calip capsule that only dissolves under specific conditions, offering a high degree of control over the timing and location of the release. Looking to the future, the field of thermoresponsive particle synthesis in microfluidic systems is poised to expand further as new responsive polymers are developed and integrated into microfluidic platforms. There is ongoing research into polymers that exhibit sharper or more tunable transitions, as well as materials that can combine multiple responses for even greater control. For example, future particles might be engineered to respond to subtle temperature variations combined with specific biomarkers, providing highly targeted therapeutic interventions or environmental monitoring solutions. These advancements will undoubtedly open up new avenues in precision medicine, smart materials, and adaptive systems. In conclusion, thermoresponsive particle synthesis in microfluidic systems is a vibrant and rapidly advancing area of research that leverages the precision of microfluidics to create smart, adaptive materials. These particles, which can change their properties in response to temperature changes, are synthesized in highly controlled environments that allow for exceptional uniformity and reproducibility. Their applications range widely from controlled drug delivery and smart coatings to environmental sensors and soft robotics, demonstrating the transformative potential of this technology. With ongoing advances in microfabrication, real-time monitoring, computational modeling, and multi-responsive design, the future promises even more sophisticated and versatile thermoresponsive particles that will impact diverse fields of science and engineering.